Infants rely on eye movements to explore the environment, especially before they're able to crawl or walk. In newborns, gaze is primarily controlled by subcortical structures, and cortical control increases over the first year. However, little is known about how the development of specific cortical regions influences gaze control. This is largely due to a lack of suitable methods. It's incredibly difficult to conduct fMRI studies with awake-behaving infants, and EEG doesn't have the spatial resolution needed to isolate individual areas of visual cortex. To address this issue, we took an analytic approach called Representational Similarity Analysis, or RSA, and used it to link infant eye tracking data to a computational model of the ventral cortical processing stream. Representational similarity analysis can be used to answer many questions about development, so we're going to stress how the method works in this video. We really hope you'll think about how you might use RSA in your own research. It's not difficult to implement, and we've provided all our analysis code on OSF. RSA was originally developed for fMRI, but it's actually a general-purpose method for assessing links amongst different kinds of neural measures, computational models, and behavior. For example, we might have bold activation across a set of voxels in fMRI, ERP waveforms from scalp electrodes, heat maps of eye movements, and a pattern of activation across the units of a neural network model. How can we compare these different sources of data given that they have completely different formats and units? RSA solves this problem by converting each source of data into what's called a representational similarity matrix, or RSM. We can then look at how strongly the RSMs from the different data sources correlate with each other. Okay, let's unpack all of this. To get a representational similarity matrix, you need to look at the pattern of data produced by several different inputs. In our experiment, infants looked at complex scenes like these. Each scene was shown for 10 seconds while we tracked eye movements. We computed a fixation density map for each scene, which just shows where a given infant looked. We can quantify how similar any two scenes are from the perspective of the gaze control system by computing the correlation between the fixation density maps for these two scenes. We get a separate correlation for each pair of scenes for a given infant. We had data from 22 scenes, so this gave us a 22 by 22 correlation matrix. This is the representational similarity matrix, or RSM, for this infant. RSMs are useful because they move us away from the methodology-specific data format of eye tracking and into a more general way of quantifying the underlying representational geometry of the gaze control system. We can transform data from other data sources and computational models into the same format, making it easier to see links between different data modalities and models. In the present study, we examined the relationship between the gaze patterns and AlexNet, a computational model of the ventral visual processing pathway. The input to AlexNet is an RGB bitmap of an image. The information from the bitmap passes through multiple layers that are analogous to the sequence of areas along the ventral stream. Several studies have shown that the lower layers of AlexNet can predict the pattern of activity recorded from early stages of the ventral stream, whereas higher layers are associated with activity in the later stages. We took the 22 images that the infants had viewed and fed them into AlexNet, producing a set of activation values across units in each layer for each scene. We then computed the correlation between the activations produced by one scene in a given layer and the activations produced by another scene in that same layer. For example, we fed this image into AlexNet and obtained the pattern of activation in convolutional layer 5. We also obtained the pattern of activation in that layer for the second image. We then calculated the correlation between these two patterns of activation. We repeated this for each pair in our set of 22 images, allowing us to obtain a 22 by 22 representational similarity matrix for this layer of the model. We now have a 22 by 22 RSM for convolutional layer 5 of AlexNet, and a 22 by 22 RSM for a given infant's fixation data. We can then compute the correlation between the two RSMs to quantify the extent to which the similarity structure of the fixation data can be predicted by the similarity structure of the computational model. This correlation quantifies the representational similarity for that infant. We then asked how the representational similarity varied as a function of the age of the infant. Here we see that representational similarity between this layer of the model and the infant gaze patterns increased with age. In other words, this layer of AlexNet predicted the gaze patterns of older infants better than the gaze patterns of younger infants. This graph summarizes the results for each individual layer of AlexNet. Showing scatter plots for each layer would be cumbersome, so here we've divided the infants into 3 age groups and plotted the mean representational similarity for each age group. You can see that representational similarity increases with age for convolutional layer 5, just as in the scatter plot. But note that age was treated as a continuous variable in the statistical analyses. You can see that representational similarity was actually higher for the younger infants than for the older infants at the lower layers of AlexNet. In other words, layers that correspond to lower-level areas of visual cortex predicted the gaze patterns better for younger infants than for older infants. This reversed for the higher layers. Layers that correspond to higher-level areas of visual cortex predicted the gaze patterns better for older infants than for younger infants. This produced a significant age-by-layer interaction in a linear mixed model. These results provide a first step toward linking the development of infant gaze control to different regions of the ventral stream. They also demonstrate the feasibility of using representational similarity analysis to link infant gaze behavior to neural network models. We hope that you find these results interesting and will consider using RSA in your own research. It's a powerful technique for linking together different types of behavioral data, neural data, and patterns of activation in computational models.